The following presentation is sponsored by the Cleveland Clinic.
The health care of the population can't improve without research into the causes of disease.
If they can't do anything for me, at least I'm helping to find and solve the problem.
Our research will contribute, hopefully, to the safe travel of future generations of astronauts to Mars and beyond.
Research allows you to take it one step further of asking an important question, but then being part of finding the answer to that question.
If research is based on results, I'm your result.
The research lab is a frontier where dedicated scientists are pushing the boundaries of medicine, finding answers, searching for cures, repairing lives. Hello, I'm Monica Robbins, and welcome to another edition of Medical Miracles. Today, we're taking you on a journey to the edge of the medical universe and beyond. From inside the Cleveland Clinic Research Laboratories to the International Space Station high above in Earth's orbit. We'll travel with modern-day explorers to places only the brightest and talented minds can go. Exploration in the research lab may begin in a beaker or test tube, but it has discovered one miracle that lies within our bodies, the stem cell.
A stem cell is a cell that has yet to be determined what its ultimate fate will be. So a stem cell can literally become any kind of cell in the body.
On a journey that began with tragedy, Elizabeth Mara learned the mystery and magic of the stem cell.
I decided to go on a motorcycle ride with a gentleman. It's the first time I'd ever been on it. I was actually a little apprehensive. I wasn't listening to my gut, which told me, you better not do this. And I just remember when he started going a little faster, I remember kind of squeezing on a little harder and squinting a little bit, and my hair was flying back. And then slowly I was realizing that there was a stop sign coming, and it just kept coming closer and closer. And as soon as I realized that we weren't stopping, I saw a car coming from this way. And by the time I thought about the car coming and we're coming towards it, collision. So I got 2 compound fractures, one in the femur, which is in the thigh, and then one in the tibia, which is in the main shin bone. And then I broke my left hand.
She was a 22-year-old college senior, looking forward to graduation, even more excited about where her journalism degree would take her. Life came to a grinding halt. After 2 years, despite the efforts of her doctor, her leg did not improve.
Month after month, it was x-rays showed no healing. I couldn't get off these crutches. I couldn't get up in the morning. I'd wake up in the morning and I'd put my leg down, and it was just this shooting pain would go through my entire body, and I didn't understand it.
That's when a friend stepped into Elizabeth's life with a newspaper article about one doctor who might have the answer. Dr. George Mushler is an orthopedic surgeon at the Cleveland Clinic. He is discovering groundbreaking new ways to use stem cells.
Some of the cells, some of these colonies, instead of making bone, may make cartilage, they may make fibrous tissue, they may make muscle. And if I change the culture conditions a little bit, I could get probably the majority of them to make cartilage, or the majority of them to make muscle.
So you could turn them into what you want them to be?
We have a pretty good ability to do that.
Dr. Mushler's research inspired Elizabeth to make an appointment.
I just had this feeling. Again, I don't believe in destiny or anything like that, but I just felt like I should go.
She wasn't disappointed. She was impressed by how much time Dr. Mushler spent with her, examining her previous x-rays. Dr. Mushler told Elizabeth he wanted a new set of oblique x-rays. Elizabeth never had this type of x-ray taken before.
She had brought x-rays to me when I first saw her, and it kind of looked like the bones were healed. But that can be deceiving, because if I hold my hands up like this, in that way you can't see any space between my hands. And then I turn my hands this way, and this way you can't see any space between my hands. But if I angle it just right, you can tell my hands are not, together. And that was the case with her bone.
With renewed confidence, Elizabeth scheduled surgery with Dr. Mushler.
this is the start of the preparation of the bone graft, is to collect bone marrow by aspiration from the iliac crest.
Dr. Mushler uses a syringe and creates one small hole to draw out the marrow. It leaves only a tiny scar.
Essentially, I've got this matrix, which is a matrix made of human bone.
The key to the graft is the bone matrix. The matrix will provide the scaffolding with which the stem cells can build bone. Dr. Mushler will fill 5 syringes with bone marrow. The marrow is then added to the matrix.
Essentially, we flood this chamber here with marrow, which is heparinized so it won't clot.
Using a new device called a selective cell retention system, the cells are trapped inside the matrix. After 2 cycles, it's time to make the marrow clot. Dr. Mushler will add a 6th syringe of marrow, plus another containing thrombin and calcium. This will help the mixture form into a gel-like substance.
The goal is to get as many of these bone-forming cells as you can into this place where you need bone to form.
As soon as the bone is ready for grafting, the matrix material is brought to the surgeon's table.
So here's our bone graft.
The matrix gel is put into the gaps of the missing bone. Now it's time for the cells to do their work.
The miracle of stem cells happens next, when they do the work of actually making bone where we need it.
Bones are not the only part of the body that can benefit from stem cells. Cleveland Clinic researchers are exploring to unlock the potential of stem cells inside the heart and eye. Dr. Mark Penn found a groundbreaking factor named SDF1, a kind of radar for stem cells. The factor helps stem cells find their way to the heart, rallies the stem cells to gather in the heart, and then help heal the damage of a heart attack.
And what we like to say is, normally stem cells get released from the bone marrow and they go back to the bone marrow. What we did was we gave those stem cells an extra address to go to. And that address, in our case, was the heart. And these cells went to the heart, they engrafted in the heart, and then tried to help recover heart function.
The eye is another place where researchers are finding stem cells repairing damage. Like a car windshield, the cornea is your view of the world, but sometimes it gets grimy and impairs vision. Limbal stem cells are like a wax repellent you put on your windshield to improve your view.
We call them stem cells because they're very undifferentiated. In other words, they still have the capacity to repopulate the surface of the cornea. The best way to think about it is like the skin. When you have a scratch in the skin, the skin grows back. That's pretty much the same analogy.
In Elizabeth's case, stem cells would answer her need. 2 years since her accident, 8 months since her surgery, Elizabeth visited Dr. Mushler for the last time.
Went in, got the x-rays, took him up to him, comes walking in, and he's just like, "You're all healed." And I just kind of looked up at him in shock for a minute. I don't think I heard much after that because I couldn't believe those words actually came out of his mouth.
I'm proud of you, though.
It was just, this big sigh, and it was just one of those things where you're like, oh, God, is it over? Could this actually be over?
It was over. The bone had grown and her leg was back to normal. Elizabeth's journey ended, but her life now has a new course. Because of her recovery experience, Elizabeth has gone back to school and changed her major. She's now studying to become a physical therapist.
I believe in science, I believe in medicine. I think if you look at my situation and you look at how horrible things were and how I really didn't think I was going anywhere, and you take me from going nowhere to everything that I want to do, then if research is based on results, I'm your results.
Next, she was losing her vision until she volunteered for a research study that literally opened her eyes. And later, how a Cleveland Clinic research project is helping to put an astronaut on Mars, when Medical Miracles continues. As you age, you have a 1 in 6 chance of developing the #1 cause of blindness worldwide. By the age of 75, you have a 1 in 3 chance. There are 2 forms of macular degeneration, wet and dry. The wet form produces an image like this. You don't lose total vision, you lose central vision, making life difficult.
I was very worried. I thought I had either glaucoma or a cataract. I didn't say anything to Leo, my husband. I didn't say anything to my daughter.
Phyllis Hill is 75 years old and loves a good book. Gradually, she began having a difficult time reading. She noticed that printed words were becoming shadowy. Phyllis contacted the Cole Eye Institute at the Cleveland Clinic and scheduled an eye exam. After the exam, she received startling news.
He looked at the chart and he said, I see your sister has macular degeneration, and we've come to the conclusion that this is what's wrong with your eye. I was very shocked by that.
Environmental factors play a large role in macular degeneration, but the physicians at the Cole Eye Institute believe that genetics may be the reason for Phyllis's vision loss.
We haven't been able to find the exact gene that causes macular degeneration, but that's some area that we're working very closely with here at the Cleveland Clinic, trying to figure out what's precisely the gene that may be causing macular degeneration, because if we could figure that out, maybe we could figure out what protein it codes and we could block it. So we are definitely looking at ways to prevent it from a genetic standpoint, but that's way in the future at this point.
When Phyllis learned she had macular degeneration, she remembered her sister's gradual path to blindness.
Probably because I had experienced the deterioration of my sister's eyes through the phone and meeting her in Ireland on some of our vacations. I saw what it did to her. She's considered legally blind, and she's in a nursing home now, and it frightened me.
Dr. Peter Kaiser was conducting a clinical trial investigating a new treatment for macular degeneration. The trial involved 1,200 patients worldwide. He thought Phyllis would be a good candidate. The study involved an injection of a drug that would block a growth factor. Phyllis would receive the injection once every 6 weeks. The clinical trial was a blinded study, meaning both doctor and patient didn't know if they were getting medication or a placebo.
Such that they could not in any way affect the outcome of the study because they would like a drug to work or would like a device to work. All of the data analysis is done in a way that the physician or scientist has no knowledge of which drug or not drug the patient was put on. So some patients are on a particular drug, others are on a placebo. The doctor doesn't know which is receiving which.
I can know they're there, but I really
The first day of the clinical trial required Phyllis to have a thorough check of her vision.
I called it a big fog. They called it a hole. Their terminology was different. It was like a black fog to me. And I could see just the outside, the peripheral vision, and I couldn't see the center at all. That's the way it was that first day.
After numbing the eye, Phyllis is given the injection. As she sits in the dark, the injection, whether it's the real medication or the placebo, has time to work. After the first 4 months of the study, Phyllis noticed a change. After 2 years, her eyesight improved.
I think she's doing superb. The blood vessel that we were treating is completely dried up. It's no longer active. There's no bleeding. There's no leakage. So hopefully it'll stay that way, and her vision has improved very nicely. So at this point, she's doing great in the clinical trial.
And gradually, good things started to happen. The fog that clouded the central portion of Phyllis's eyesight had lifted. Dr. Kaiser doesn't know whether or not Phyllis received the drug, but the results have been remarkable. Regardless of what she received, Phyllis went from being legally blind to a remarkable 20/40.
What's the future of macular degeneration? Can it be eradicated?
Hopefully, over the next 5 to 10 years, by using combinations of these drugs as well as the new drugs that are being developed, we can cure macular degeneration. I think in 10 years we will cure macular degeneration.
Phyllis and her husband Leo felt blessed when Phyllis joined the clinical trial.
It's a miracle because it nearly knocked me off my feet. I wasn't expecting anything as dramatic as this. I always thought when your eyesight goes, your eyesight goes. And to know and see the improvement and then have the doctors verify what I'm feeling and what I'm experiencing, it's a wonderful, it's a miracle. It is a miracle. And that's why I think medical miracles are there. And I'm very thankful for it.
When we come back, the manned mission to Mars may not launch unless scientists stop space travel from weakening bones. We'll meet the Cleveland Clinic doctor who put his research to the test in space. For centuries, humans have fantasized about a voyage to Mars. This year, NASA dedicated itself to putting an astronaut on the red planet, and the Cleveland Clinic is helping to make that adventure a reality.
We leave as we came, and God willing, as we shall return.
The last moon mission was a stepping stone to the future. The next giant leap for mankind will be taken on the surface of Mars.
That'll probably be the greatest adventure in spaceflight to this time, because up until now, while certainly there was a lot of risk there throughout the years in comparison to most things, you were never far away from home. I mean, if something happened, you could come back. Even in Apollo 13, they're only a few days from home. When you go to Mars, you know, you can be a year away from coming back.
Because of the danger, an astronaut must be at peak performance. One roadblock to success is the rapid bone loss astronauts endure in weightless environments.
A typical stay, long duration stay in space, is equivalent to 7 years of aging. So, in fact, that's bone that you've got to get back, equivalent to having just done a normal life of 7 years on Earth. And so it is a very significant loss.
For an astronaut, the bone loss rate is about 10 times faster than the bone loss occurring in a postmenopausal woman. The dangers of weak bones are a serious concern.
In the process of doing what is quite normal here on Earth, fracture a bone, and we don't know how that bone's going to heal. So I think most authorities would say we are not yet ready to go to Mars, but we better be within 5 to 10 years.
The trip to Mars will be a long journey that will take its toll on an astronaut's body. But it's on the surface of Mars and the return to Earth where gravity will put the astronaut's weak bone to the test. Dr. Peter Kavanaugh of the Cleveland Clinic was inspired to find better ways of measuring bone loss in space and help in the quest for Mars.
The issues are really these. When you get to Mars, which has a gravity of about 1/3 that of the Earth, how are you going to arrive with healthy bones and keep healthy bones such that you don't fracture your bones during a normal period of work and construction that will be actually carried out on Mars? So NASA's goals to explore our near part of the universe with healthy people impinges directly on the work that we do.
94, 99.
Dr. Kavanaugh's team had to perform the experiment on Earth before it went into space. The difficult part was reproducing the same effects of space travel. In order to do so, the team had to invent a simulated weightless environment.
We have devised a method where we hang people up. They're looking at the ceiling and they're lying horizontal, and we attach constant force springs to all of the major body parts so that the person actually feels weightless. We then accelerate them to an exercise device using the kinds of bungee cords that are used on orbit, and so we can actually simulate exercise as it occurs in space.
Yeah, this sucks.
Astronaut Ken Bowersox was assigned to carry out the research on the International Space Station.
Microgravity environment is the advantage of the experiment, too. It's what makes it different from an experiment on the ground and why we want to be here in orbit.
Dr. Kavanaugh's experiment is designed to measure the differences between the load that the legs experience on Earth and the load they experience in space. These measurements have never been done before.
So the astronaut pulls on these pants. They look
To accurately calculate the measurements, Dr. Kavanaugh invented the lower extremity monitoring suit. The suit measures 3 classes of variables. It measures the forces under the feet, the rotation that is occurring in the 3 important joints of the lower extremities, and the suit measures muscle activity. Electrodes on the muscles record the tiny electrical signals that the brain sends to the muscles, and they can do this for both the arms and the legs. Ken Bowersox performed various exercises wearing the suit. Riding a bike, lifting weights, and running on a treadmill were all necessary tasks in order to obtain the data Dr. Kavanaugh needed.
So what does it mean to those of us on Earth? In a word, osteoporosis.
Osteoporosis is a deterioration of the skeleton to the point where very simple trauma can actually cause the bone to break.
Osteoporosis is the most common metabolic bone disease in the United States. More than 10 million people have it. 80% are women.
And about actually 1 in 6 women will have a hip fracture before they die. And if you live to the age of 80, 1 in 3 women will have a hip fracture.
What helps fight osteoporosis? Besides milk and calcium supplements, exercise is vital to keeping healthy bones. But not just cardiovascular exercise. weight-bearing activities, including aerobics, walking, running, tennis, anything that involves the force of gravity applied to the bone.
We will learn from our studies in space. Eventually, we will bring the exercise level up to threshold that is enough to prevent bone loss. And we will then be able to come back and apply that on Earth and say, we now have learned how much exercise is needed to prevent bone loss. And I think that can have great relevance to people who are trying to have a balanced program of osteoporosis prevention, which would include exercise, diet, and perhaps drug therapy.
Like the adventurers of old, astronauts will head into uncharted territory. Researchers like Dr. Kavanaugh are helping to map out a safer route.
We are now, as a nation, have resolved to send people to outside low Earth orbit. We haven't done that for many years, but now we have decided to do it. To do that, we must learn how to keep these individuals healthy. So our research will contribute, hopefully, to the safe travel of future generations of astronauts to Mars and beyond.
Medical research will affect your life now and in the future. The dedication of scientists to push the boundaries of knowledge will someday create solutions to things we never thought possible.
There aren't new oceans, there aren't new forests, there aren't new jungles to explore, but there's certainly an awful lot about nature that exists in biology that is important, is interesting, and is particularly gratifying because it can be translated into real benefits for people.
It felt so good to have somebody really want to help and really want to fix me, and to know that they could.
I'm looking at all these beautiful children, and grandchildren, and great-grandchildren, and saying, I've got to do this. Because if they find out one little thing, over the years, if their time comes that they have to face this, maybe they'll have the solution.
What's exciting for a laboratory investigator is that there are answers every day. They're not always the answers you want, they're not always the big answer, but they're pieces of information you're gathering that you eventually put together, not just you, but the other scientists in the field, to make a story. So you're gathering pieces of the story and hoping at some point it will all come together and lead to a great discovery.
If you'd like more information, log on to WKYC.com or call 866-819-8326.
Nice deep breaths in and out.
It felt like someone was choking me, and you cannot breathe.
Jennifer Duncan doesn't like to think about her freshman year of college. She spent most of it fighting an unknown illness.
I was just so sick, and nobody knew what was wrong with me. I mean, I fractured a rib from coughing.
She spent 2 weeks in the Cleveland Clinic.
They didn't know if I was going to make it. That's how sick I was.
Doctors finally realized Jennifer had asthma. Her disease is under control thanks in part to a new clinical device. How well she breathes doesn't always warn her of an oncoming attack.
Levels are high today.
The level of nitric oxide in her breath provides those answers.
When I'm very sick, my nitric oxide levels are really high, and I can adjust my medications.
Traditional asthma tests just measure lung volume. Nitric oxide measures inflammation, a cause of asthma attack. But Cleveland Clinic researchers want to measure other elements in Jennifer's breath to see if they too can be markers for lung disease.
Some of those are things like carbon monoxide that you exhale.
volatile organic compounds, oxidant molecules. These things can be elevated or reduced in the presence of lung diseases, so we're trying to figure out what normal levels would be.
The nitric oxide discovery was instrumental in helping doctors understand asthma. The next goal is to widen the use of the test.
Try to detect the disease early. Maybe if we find somebody that has high NO levels, it may be an indication that they have asthma.
Right now, Jennifer checks her NO levels 3 times a year. Someday the device may be small enough that she can check every day at home. Monica Robbins, Channel 3 News.
This is the signal ratio.
In a lab tucked in the corner of the Taussig Cancer Center is a group of scientists searching for answers in the rarest of blood diseases.
Regardless how frequent it is, it affects life of people.
Dr. Jaroslaw Maciejewski leads this group of young minds because in the commercial world there is little interest finding cures for diseases that affect just a handful of people. But there is real reason to learn.
Some very rare diseases have incredible importance to the understanding of how organs work or how other diseases evolve.
They are surrounded by some of the latest high-tech research equipment, but never forget that in this lab, every test tube is a real patient.
We are dependent upon patients, and there is a human factor involved.
They have the ability to take the answers they find directly to the bedside, but it's the questions that keep them going.
How long am I going to live? Is it going to help? What are the chances that this drug versus the other is going to help? And you know, the more you say, I don't know, the more you get a drive to do something about it, to know, to learn.
It's in places like this where the small steps are taken to reach more than solutions.
So it's not only discovering the cure, it's also improving what we do.
Monica Robbins, Channel 3 News.
So how are you doing?
10 years ago, English professor Susan Huff learned she was in danger of losing her ability to read. Glaucoma was stealing her eyesight.
I've lost significant amount of peripheral vision in the right eye, so I have straight-on vision, and it's the silent disease. It doesn't really tell you that it's happening.
There are several new high-tech instruments meant to detect glaucoma before the damage is done, but do they work?
I hope your eye's feeling more comfortable.
Dr. David Huang is leading a multi-hospital study that will track 800 people with and without glaucoma, and some who are suspected of having the disease.
We're going to see if the changes in the eye that's detected on these imaging tests predict visual loss in the future.
It would also help with treatment decisions.
What we want to do is to measure the structure of the optic nerve and the tissue around it very precisely with imaging tests to measure the progression of glaucoma.
Primary open-angle glaucoma is the most common form of the disease. Over time, the eye's draining canals clog. Eye pressure rises because fluid isn't draining. That pressure gradually deteriorates the optic nerve, causing vision loss.
We have to prevent it from progressing because we can't fix it once it has progressed.
If you have a genetic history of glaucoma, advanced imaging may be able to diagnose it earlier. The goal of the 5-year study is to establish criteria to better measure the progression of glaucoma or the risk of it. Back to you. Joe Turry was due for his annual colonoscopy when the Cleveland Clinic asked if he'd take part in a clinical study.
I almost didn't do it because I thought, I want to do a colonoscopy. I'm not going to do this other nonsense. And I thought, well, yeah, anything for mankind. I told them, I said, you know, anything for mankind. If it'll help mankind, I'm there.
Just bring your arms above your head.
He had no idea he was the kind of man it would help.
There was no indication whatsoever that there could be something wrong.
The study is comparing the accuracy of virtual colonoscopy or colonography using a CAT scan to standard colonoscopy. Preparation is similar, but the scan can see more.
The beauty of virtual colonography is that things outside of the colon can be examined, all of the solid organs.
Joe's results showed something deadly.
When I looked at the CAT scan result, I was shocked because the radiologist suggested that there was a malignant tumor or cancerous tumor within the kidney.
Dr. Carol Burke was the first to reach Joe and tell him to return for a kidney check.
And thank God she insisted, because if she hadn't, I'd have been in trouble.
His doctor told him the tumor had to go.
I said, the mass has to come out? He said, no, the kidney has to come out. Because 99% of the time, if there's a tumor in a kidney, it's usually malignant.
The remarkable thing about Joe is he had absolutely no symptoms. And he was in the study just because he thought it would be for the good of mankind, and it turned out that this cancer was detected.
Little did I know it was going to help me in the end.
His surgeon removed the kidney laparoscopically with a very small incision. Joe was home the next day.
And the beauty is that the cancer was in very early stages, so Joe was cured.
So the study literally saved his life.
It saved his life.
Now this will simmer for an hour and a half.
It's never been done before, and it's really sharp. It's the very first step toward...
Dr. Emil Paganini is part of a research team working on the world's first bio-artificial kidney. It doesn't look like a kidney, but it has the ability to act like one.
What we're doing here is really the first step towards incorporating living cells. So it's almost as if you had a kidney transplant, but it's in a halfway house, if you will.
The research is still in its infancy. Only 10 people with acute renal failure have tested it, and only for 24 hours. But early results show the potential is endless.
I believe this is the first step towards an implantable bioartificial kidney. That would avoid us having to worry about having donors, but rather we would be able to perhaps even take your own cells.
It works in conjunction with dialysis, the standard filtering treatment for those with renal failure. When the kidneys fail, the body can't eliminate toxins. Inside the kidney, cells begin to die. The dialysis machine filters toxins from the blood. In someone with acute renal failure whose kidneys are not permanently damaged, the cells may begin to regenerate, healing the kidney. But that takes time. The bioartificial kidney contains living human kidney cells taken from a donated organ that for whatever reason can't be used. Researchers incubate them and grow billions more. Then they're injected into this plastic tube. The cells begin to grow inside these hollow hair-like strands. After dialysis, the filtered fluid then passes through this tube outside the body, bathing the cells. The cells then charge the fluid with information signals that get sent back into the bloodstream and back to the ailing kidney.
They're much smarter than we are. That's what they do for a living, is clean this fluid. And so they're able to do whatever it is that they do. And by doing that, it sends signals back to the kidney, signals that we don't know about yet, to tell the kidney to clean up its act, if you will.
Causing the kidney to begin regenerating its own cells and heal.
The kidneys themselves, our whole body is unbelievable in what it can do. And I think we are appreciating that now, and we try to replicate that.
Paganini believes the signals travel to other organs too and boost the immune system. Future research may make it small enough to implant in those with chronic kidney failure.
And that would avoid this long wait for transplants.
Paganini estimates more than 500,000 people have acute renal failure, making up nearly 10% of all hospital admissions. Now, with the aging baby boomers and the diabetes epidemic, this groundbreaking research could be extremely vital. Engineering designed the simple plastic tube and strings. Genetic and cell research made this possible.
Putting all of that information together and putting it into something that would actually help somebody is, to me, one of the most exciting things that medicine can do.
Monica Robbins, Channel 3 News.